So like Dr. Hammer said, my name is Trayce Blas. And so kind of some background behind this research. I originally got interested in this research, like he said, taking experimental plant science and also taking environmental geology at the same time. So plant science kind of introduced me to the phytoremediation aspect of it, whereas environmental geology introduced me to just bioremediation in general and the remediation of like nuclear materials and how we dispose of those. So that's kind of some background of how I got interested. I started talking with Dr. Hammer, and around that time is when I needed to start like thinking about my honors project and what I wanted to do and what I wanted to research. So I chose the current understanding of the phytoremediation of radionuclides. So on the agenda today, so we're going to start off just what's the purpose of my research, what was I researching. We're going to move into what exactly a radionuclide is for those who aren't as familiar with scientific terminology. Then we'll move into what phytoremediation is in general and talk about the different methods like phytoextraction, sequestration, filtration, volatilization, and degradation. Then we'll move on to some aspects that affect the remediation efforts. We'll talk about the treatment of post-remediated biomasses. Then move on to future prospects about what this kind of technology will look like in the next 10, 20, 40, 50 years. And we'll wrap it up with some conclusions and leave some time for some questions. So the purpose of my research was just to understand the different processes used in phytoremediation, understand the different, like, specifically plant processes, like their metabolism, hyperaccumulation, things like that. I also wanted to determine what kind of plants and what areas phytoremediation would be possible. And then to determine the future prospects of this technology. Like I said, what it will look like in 50 years from now. So let's start off with what exactly a radionuclide is. So a radionuclide is an unstable isotope of an element that emits radiation as it decays. So typically it happens in three ways. There's gamma radiation, there's beta decay, and there's also alpha decay. Alpha decay and beta decay emit respectively alpha particles and beta particles. And gamma decay emits photons, which are particles of light. These typically are high energy photons. Think of like gamma rays, X-rays, things like that. Things that can cause damage to living tissue, thinking like cancer, for example. And so where you might see these or typical uses, obviously medical equipment uses them, your PET scans, your X-rays are going to use them, a lot of different scientific equipment uses them. There's also nuclear weapons, obviously used for certain isotopes of uranium, plutonium. Also nuclear reactors. Nuclear reactors use specifically uranium-238 most of the time. And we'll see incidences where problems happen with nuclear reactors, like the Chernobyl nuclear disaster in 1986 or Fukushima in 2011, spreading radioisotopes into the environment. And also mining of naturally occurring radioisotopes. So things like uranium-238, 235, thorium-232, potassium-40, all naturally occurring in the environment. So, moving on, phytoremediation in general is the use of plant processes to clean, remediate soils, getting those pollutants out of the soils, or just environments in general. So the diagram kind of provides a just brief overview of each individual process. We'll go more in depth in them. But the exact processes are phytoextraction, phytosequestration, phytovolatilization, rhizofiltration, and phytodegradation. So moving on for phytoextraction. So this is the use of the roots of the plant to absorb these contaminants and translocate them into different aerial parts of the plant depending on the environment of the plant, soil conditions, etc. So these may be like stems, your reproductive structures, your leaves, your shoots, depending on the type of plant. And so it can, the diagram below shows like the different potentials it has. So typically you're dealing with heavy metal soils, and that can be found in, it lists a variety of ways, areas surrounding industries and manufacturing plants. And so when dealing with phytoextraction, you're looking at certain characteristics of the plant. So for phytoextraction specifically, you're looking at the root system of the plant, how large is the root system, what kind of area will that affect, and also the biomass of the plant. So how much mass does the plant actually have, and where will these pollutants be concentrated on? So examples I found were like corn. Corn showed some potential, sunflowers, Indian mustard. Probably one of the most common examples I found was vetiver grass. So that's native to Africa. And it has showed great potential for hyperaccumulation of specifically uranium. So in my studies, I found that concentrations of soil with 927 parts per million of uranium, after 60 days, it removed 90% of the uranium in that soil. Getting to concentrations greater than 1,000 parts per million showed actual movement of the uranium into those aerial structures. So what scientists believe are happening here is as the uranium enters the plant cell, it's actually causing issues with the plasmodesmata, which is how plant cells transfer things from different plant cell to plant cell. So when that is damaged, it remains permanently open, and that allows for uranium to be transferred into the xylem, and that then translocates it into other parts of the plant, like the leaves, the stems, the reproductive structures. And so these contaminants can have deleterious effects on plants, and sometimes it isn't beneficial, and sometimes it is. Moving on to phytosequestration, this is all about stopping the plants from continuing to contaminate the environment and spread to areas like groundwater, for example. So there's three main processes this can happen. It's the use of phytochemicals is one of them. Phytochemicals are chemicals actually produced by the plant, and so it can cause the precipitation of pollutants within the soil. There's also certain root membranes of plants that allow for the permanent binding of these pollutants directly to the channel proteins on the membrane. And then they can also be sequestered in individual plant cell vacuoles. There's an example of the vacuole there. Those are typically for like waste storage or storage of water. And so some plant cells will purposefully absorb pollutants and transfer those and sequester them in the vacuole itself. As in terms for research of radionuclides, there wasn't much out there that I found personally, at least current studies. So that's something that still needs to be studied. I'm sure there's plenty of opportunity for potential plants to do this process. Moving on to phytovolatilization. So this is volatilizing these chemicals, turning them into gas, and then releasing them into the atmosphere. And so this can happen in two different ways, a direct pathway and an indirect pathway. In the direct pathway, these pollutants are absorbed through the roots of the plant and then translocated into the leaves, which then turn them into a gas, and they get released into the environment. So that requires the direct contact of the plant and the radionuclides, or the pollutants. The indirect pathway involves certain root activities, so the roots may cause the pollutants to volatilize, or they can lower the water table. So gases actually moves easily through air more than it does through water. So lowering the water table allows for a greater amount of unsaturation of the soil. There's going to be less water in that soil. That's called the vadose zone. So lowering the water table increases the vadose zone and allows for greater release of the volatilized pollutants into the atmosphere. So there's a couple examples. Starting off with Fukushima Daiichi nuclear power plant, I mentioned this earlier. That incident occurred in 2011. Tsunami caused by an earthquake crashed into the power plant, causing core meltdown. Nuclear engineers attempted to stop the core meltdown by pouring water into the core to keep it cooled. And in that process, they created tritium-contaminated water. Tritium is an isotope of hydrogen, hydrogen-3, and it is radioactive. So the current Japanese government plan is to slowly release this tritium-contaminated water into the Pacific Ocean. There has been some fight back from local communities, neighboring nations, about worries about biomagnification or harming just the ecosystem and the food chain in general. So the potential use of phytovolatilization here, or other even phytoremediation technologies, is great. Also, the Savannah River site in South Carolina. This is an area where there was a production of a lot of nuclear materials, a lot of radioactive materials. What they're doing there is they're actually, you can see in the photo, there's a pine tree forest around the site. They're irrigating the tritium-contaminated water into the pine trees and allowing the pine trees to actually volatilize that water and release it into the atmosphere, where it can be diluted and eventually it'll decay into helium. I think the half-life is like 12 and a half years. Yeah, doing that in the past 21 years, they've reduced their amount of tritium on site to 70% of what it was originally. Moving on to rhizofiltration. Rhizofiltration is very similar to phytoextraction. It's using the plant roots to absorb things, but specifically in aquatic environments. So this could be your ponds, your lakes, your rivers. In the lab, hydroponic solutions. For those who don't know what hydroponics is, it's an alternate way to grow plants, but rather than like traditionally growing it in soil, you're planting it in like a water medium. So it's a watery solution of like nutrients, minerals, anything the plant needs to survive. And so sunflowers, annual sunflowers, are actually a common example of use for hydroponics in rhizofiltration. Also, duckweed is pretty common. Duckweed would be a good choice as well because it grows relatively quickly and it's naturally occurring in water. So it has the root system and the ability to support life in the water. Next, we're going to move on to phytodegradation. So this is the use of actual plant metabolical processes to remediate these pollutants. Typically, it involves chemically altering it into a less toxic form. This has limited application to radioisotopes, as you can't really chemically alter a radioisotope because that would have to take place at the atomic level. So that's where radioactive decay comes in, where you're adding and getting rid of neutrons, protons, things like that. So in terms of aspects that affect phytoremediation efforts, obviously cost and maintenance are going to be a big thing. So typically speaking, specifically for phytoextraction, where you're actually harvesting the area of the plant where the pollutants are concentrated, that obviously has cost to it. Shipping is off to the facility, off-site, for further treatment. And obviously in more developing countries too, they may not have that cost. Obviously plant growth, the duration of plant growth is a big factor of this. Some plants grow faster than others, some plants grow in different climates, as I'll mention later. Yeah, and obviously climate's pretty common. You're not going to grow a plant that's native to like Nebraska and Siberia, and plants that are native in Ukraine where Chernobyl is, you're not going to be able to grow exactly in like the area surrounding Fukushima. Also, soil viability and availability. Phytoremediation obviously will have like limited efforts in city areas where there's a lot of infrastructure and not a lot of available soil. There's also pH of the soil. I read quite a few articles where pH can actually reduce the ability of hyperaccumulation. I've also read certain soil amendments like potassium, phosphorus can also affect its ability to hyperaccumulate. So, moving on to treatment. So, there we go. Okay. So treatment. So after collection of the biomass, that's called post-phytoremediated biomasses. It's the collection of biomasses that have the pollutants concentrated within it. So the most common one is incineration. That's the controlled burning of these biomasses. It's reducing the volume of the biomasses is kind of the main goal there, picking out specific radioisotopes, which then can lead to long-term storage. So typical storage, typically for like high-level nuclear waste, you have the dry caskets above ground that they keep an eye on, or geological repositories storing them underground. And something I added recently was industrial application. Some research I found not too long ago, within the last 2 weeks, that I decided to add was a news article over the possibility of nuclear diamond batteries. So it's using the heat produced from radioactive decay of these radioisotopes, and they're using that heat to create electrical energy through, I think it's diamond semiconductors. So that's some relatively new information that I actually just found out about not too long ago, and I'm curious to see where that goes in the future. I know the article discussed using it for like aeronautic applications, so like your smaller devices used in aviation or space travel. They also mentioned like even like commercial use of like powering our watches that don't require much energy. It's more for things that don't require a lot of energy but require energy over long periods of time. Talking about future prospects now, biology, the new big thing in biology is biotechnology, using genetic engineering to alter the genome of plants and animals. We see this a lot in agriculture with BT corn having natural resistance to pests like the European corn borer. We also genetically modify livestock sometimes. And so the big new thing with this is the CRISPR-Cas9, which essentially acts as molecular scissors to edit a plant's genome. So this could lead maybe to increased plant endurance, potentially editing the growth of plants and how they grow, maybe altering metabolic processes. The ultimate question is going to be, is this going to affect the use of it? Are we going to see an increased use of it, or are we going to see a decreased use of it depending on like the cost it takes to genetically modify these organisms? So big questions on whether or not that will increase the use of this or not. Finally, some conclusions. So phytoremediation has shown to be a cost-effective and more environmentally friendly way to remove pollutants from the soil in general, better than like chemical treatments or whatnot where they have to be shipped off by truck. It's also a wide variety of plant species might have the ability to use one of these methods, extract, sequester, volatilize, or filter pollutants. There's hundreds of native species to even Nebraska that have been unstudied, so there's a lot of potential study for phytoremediation, which plants may be used depending on the different processes that you are looking at. So thank you guys for coming, and I'd like to open the floor to questions if anybody has any.